Hey everybody, Jake Thorne here with Texas A&M AgriLife Sheep and Goat Extension. So if you've been following our Facebook or YouTube videos, you know that we are big into capturing parasite resistance amongst small ruminants, sheep and goats here at the research station. So one of our previous videos also explained a challenge that we have going on here where we artificially infected some animals, some sheep, with Haemorrhagis contortus larvae. And the goals of this project are to capture the variation in various measurements of the way that these animals handle this parasite challenge, and then we're actually going to compare that against their DNA profile. So we use fecal egg count, which is something we've covered before, also FOMACHA score, which is where you look at the third eyelid of the animal for a measurement of anemia. But in this project, one of the biggest concerns and one of the things that we're really trying to focus on is also animal welfare. And so one of the ways that we really ensure that these animals aren't showing too harsh of signs from this parasite challenge is by capturing a pack cell volume or hematocrit. And so this actually relates to the FOMACHA score. FOMACHA scoring is really just an estimation of anemia, but through this method, we actually capture anemia directly. And so what we do is we capture whole blood from the animal, and then we load that into a capillary tube, which is shown here, this little tiny tube, and we load it into this microhematocrit centrifuge. And what this does is it separates the components of the whole blood between the red blood cells and everything else. And the percentage of the red blood cells is what we're interested in measuring. So these animals started, they were on full feed, they were completely healthy, and their average hematocrit or PCV was 40%, which is really, really healthy. Now, as this challenge has progressed, which we capture this weekly, that started to drop down a little bit each week. And so anything below 20 is when we are going to basically treat that animal and get rid of those entire internal parasites within that animal so that there isn't any harmful effects. But at this point, our animals are still fairly healthy. The average PCV is still in the low 30s, which is very good, and it still allows us to keep capturing fecal egg counts and FOMACHA scores to determine which of those animals are potentially resistant and which may be more susceptible to Haemorrhagis contortus. So this is just one more tool that allows us to determine variation. It allows us to monitor safety of our animals throughout our projects, and it's really important and something that we're really focused on, particularly in this artificial parasite challenge. If you haven't seen those videos, I encourage you to go back. This is actually week 6 of this project. So 6 weeks ago, we posted a video on the day it started. It showed us inoculating those animals and explained it a little bit further. I encourage you to check that out if you haven't done so. Thanks and have a good day.